So I am, as Keelan said, I'm Gabe. I'm a postdoc in the Fortin lab, and I just want to thank the organizers that even though Norbert is unable to speak with us today, and as sad as that is, because you now have to listen to me present this, we're really happy to be able to share this work and to get it out there for everyone to be seeing. So I'm going to be talking to you today about the work that we've been doing for quite a while and looking at CA1 activity as animals perform a non-spatial sequence memory task. I just want to say from the outset that this is a team effort. Neither Norbert nor myself are data scientists, but we work with an amazing group of people who are. The majority of the work that I'm going to be sharing with you today is the result of this collaboration with them. Just wanted to make sure I gave them a shout out here up at the top, just in case we get interrupted by a squeaking chicken at the back end of the talk here today. So we know that our memories are organized in space and time. This is a point that's been made before. It's come up a couple of times so far. What we're interested in the Fortin lab is understanding how we remember an event, how we remember events. We remember events, they tend to be remembered when and where it occurred. Fortin lab, we're mainly interested in this latter feature. So quick example from, Norbert's daily life. That is the order of Zoom meetings that he has to put up with throughout the day. And so what we're interested in studying is how these events are organized in time and how the brain can kind of discriminate these events based on when they happen in the sequence. Now, I'm going to be talking to you today about work in the hippocampus, but we know that the hippocampus isn't the only region for this today. It isn't the only important region for this. Dr. Tambini did a very good job of showing us some other regions this morning that were involved. But we think that hippocampus is serving a specific role, and that is to encode and preserve the sequential order of experiences. And that these representations enable us to predict and use that information to inform decision making. So a quick organization of the results for today. I'm going to talk to you a bit about the approach that we use to do this work. And then I'll talk to you about some temporal coding that we find in CA1 ensembles, how we can identify trial-specific information, and how we see sequential reactivation of representational content throughout individual events, and how, more or less, this information can then be compressed even further down into a theta cycle. So let's jump into cross-species stuff, because I have a lot of stuff I got to squeeze in today. So this work began as a collaboration with the Stark lab here at UCI, and we're hoping to be continuing on with it with the Bornstein lab. And the main structure of this task is that subjects have to discriminate items based upon when they occur within a sequence of items. For rats, we use a set of odors, all presented from a common port. In the human version of this task, we use a series of abstract images, all presented at the same location on a visual screen, on the computer screen. Now, the key manipulation here is that all of these items are presented at a common location. This removes spatial cues from the equation and ensures that the behavior is really dependent upon the temporal features of these items. Also, from a cross-species perspective, it works out pretty well because human imaging tends to benefit from the subjects being stationary. So let me dig into a little more of the specifics, but I'll go through it pretty quickly. In this task, rats are presented with a series of odors. The sequence itself evolves over multiple trials, such that only a single odor is presented on each trial. The rat pokes in at the beginning of the trial and presented with an odor. If it's in the correct position, they have to discriminate whether the odor is presented in the correct sequence position or not. If the odor is in the correct position, meaning that A is the first trial, B is presented on the second, and so on, they have to keep their nose in the port for a little over 1 second until they hear a little beep that signifies they held long enough and they can withdraw from the port to get their water reward. If, for example, one item is presented out of sequence, and we only present up to one item out of sequence, that way we know what sequence they're using. If one item is presented out of sequence, they have to withdraw from the port before when that beep would have occurred. Now, with the human version of the task, they have to hold down a key on the keyboard, and we allow them to use their fingers instead of their nose. some quick rapid fire results from this approach, we find that the behavior on these tasks is remarkably well matched in rodents and humans, meaning overall performance is pretty comparable between species, as well as the kinds of mistakes they make are pretty much the same. Moreover, regions that we know are involved with that need to be online for the animals to perform the task accurately are likewise engaged when humans are performing this task. What we've shown previously is that CA1 activity in both single cells and local field potentials is capable of differentiating the temporal context of these items, meaning whether they're presented in sequence or out of sequence. So today, what I'm going to be showing you is an extension of this work, just demonstrating that there's a massive amount of temporal and sequential information that's present in the ensemble that you can decode using fancy statistical models. So now that we're done with going over the approach, let's start talking about where the temporal information comes in. To investigate temporal coding, we're using a Bayesian decoding algorithm that has been kind of adapted from spatial work. What this does is this algorithm is able to decode an animal's spatial location based on the activity in the ensemble. For spatial decoding, you're looking at place fields, the place cell activities. In temporal decoding, the way we're applying it here, we're looking at more of the temporal fields for these cells. And so what I'm showing you here is our temporal field analogs, which is just the peristimulus time histogram for each cell. So this is the normalized average activity for all the cells in our ensemble, average across time during the different odor presentations. And they're being sorted here by their latency to peak firing. What we observed was that there was strong decoding, temporal decoding. That is, the observed time was reliably well matched to the reconstructed time, and that was associated as the highest probability throughout the trial. Interestingly, what we find is that the organization of these temporal fields within the ensemble was odor specific. And you can see this this becomes very obvious if you take the activity during a different odor and sort it during one odor and sort it by the activity on another. So for example, here, this is B, C, and D sorted by the latency to peak firing in A. You can see that these nice sigmoidal diagonals are no longer present in these other places. This isn't something specific to organizing by A. You can see this if you mismatch the organization and the activity. Those of you with a keen eye may notice that there are some differences going on here. And one of the things that we saw is that when we take the activity during one odor and use the algorithm trained on a template of a different odor, the accuracy of the temporal reconstruction scales with how far apart those two are. Meaning that if you are taking the template for odor A and decoding activity during D, you're much worse at decoding the time than you are if you were looking at the activity during B. So this was interesting to us because it tells us that while the ensemble dynamics during the trial are odor specific, there's some underlying feature that's relating them as a function of their position in the sequence. And so we thought that one thing that could be doing this is if there was some sort of long scale time series or temporal code that was evolving over the entire sequence. And that is precisely what we saw. Now you may notice here that these plots aren't as impressive as the ones from before, and that's because I'm only showing you the activity of one rat here. Because this task is self-paced, the timing of the individual trials during the sequence varies from rat to rat. And so we can't really collapse here to give you an average perspective of an average picture of what the data looks like. Even so, with these limitations, we were still able to accurately reconstruct time at higher than chance levels, so that it was significant relative to permutations. Collectively, what this tells us is that CA1 displays distinct ensemble dynamics that could be providing temporal information about passage of time within as well as across sequential events. So looking forward with this, with using our task and looking at temporal coding, we wanted to see, well, is there information about the specific events that are going on here? Is there something about the different information that the animals have to use to perform this task accurately. And to do that, we turned to an unsupervised learning approach, an autoencoder. Without going into the specifics, because I can't, this is a nonlinear analog to principal component analysis in that it allows us to compress high dimensional activity into more informative and manageable dimensions. It's unsupervised in the sense that we're not providing any labels to it. We're not telling it what the data is that it's seeing. The whole purpose of this network is to take the input layer and recapitulate it as the output. So the training of this model is to make it so that it's able to recreate the inputs on the output. The thing about this architecture that is unique is that the activity is flowing through this bottleneck layer here, where it is literally 2 nodes. And we can use the activity of those nodes to define a projection space where we can visualize the organization of the data. So in somewhat more technical terms, it's creating an unsupervised latent space that defines the structure of the data. What we found was that information about the odors, their temporal context, as well as the trial outcome are all differentiated during the trial, but they peak at different points. So what I'm going to be showing you, what you can see up at the top here, this is the example latent space for one single rat. Again, just like how the self-pacing of the task means we can't average across the entire sequence across rats, the latent space that's constructed by the autoencoder, as well as other models I'll show in a minute, they are very specific to the data that we feed it. It's individual animal specific. So the overall accumulation of that is how well we can decode that particular piece of information we're asking about. And that's what we can collapse across animals. So this is an example of the decodings and the embeddings. And what you can see at the start of the trial, this basically just looks like a Gaussian cloud. And even so, on average, we can still decode the identity of the odor at levels greater than chance. So this chance level is here is determined, it's the 95% confidence interval. It's determined by permutations. But importantly, the odor information seems to peak a little bit later in the trial, and it's remaining high, and it's staying above chance throughout the whole trial period. In contrast, information about the temporal order, meaning whether the item is in sequence or out of sequence, it's above chance at around the time that the odor information starts to peak, but it really doesn't become highest till late on in the trial period. The information about the trial outcome, as you might expect, peaks at the end of the trial when the animal, after the trial ends, when the animal is receiving its information and it's getting its results. But there is some information about whether the animal is going to be successful or not that we can decode before the end of the trial period. So notably, the order of these information sources peak in the sequential relations that we would expect given the serial information processing necessary to perform the task. The thing that's interesting, that interested us the most was this sustained odor differentiation as well. You can see it's actually even before the trial starts, there's some differentiation occurring. So that's what we're going to be digging into next. To do this, we're following up our observations. We're to dig into the content of the odor information. What we're doing is we're using a convolutional neural network approach here. Now, this is a supervised learning approach now. We're telling the model what the data is, and we're training it on a time period that starts at the beginning of the trial, the 150 to 400 milliseconds after trial start. This kind of neural network is used largely in image processing, and it's used to define spatially invariant features to aid categorization. In our case, we're adopting a tetroidwise architecture for the model to try and identify temporally invariant features present within the ensemble dynamics to evaluate, so that we can evaluate representational content. So this model is being trained at the beginning of the trials, as I said, and we're then applying it throughout the rest of the trial period. Also quickly, it's including both spike and LFP information. The LFP, it can work on spiking alone, but you get some benefit from including the LFP. Similarly to the autoencoder, we can define a latent projection space for the data here. And in this case, we can use that space to examine the decoded probabilities within this space to apply boundaries to it associated with different odor representations. And so for just as an example, you can see, and again, I'll mention the boundaries and the latent space I'll be showing here up at the top is from one specific animal. The organization of this space is very animal specific, so it's unique. This definition is unique to the animal I'm showing you as the observation. The decoded probabilities I'll be showing below will be the group averages. As you can see, there's not a lot of separation in space. We're looking at a time point well before the start of the trial here. But as you get closer to the trial period, what you start to see is that the activity on trials tends to cluster into this area of space reflecting B. And as you move forward in the trial period, it seems to transition over towards the C and D boundary, towards the representational area for odors other than what's currently being presented. When we look at the group averages, we see that this is replicated across the group, namely that odor B is decoded before the start of the trial and at the start of it, but that the decoding for C and D, these later points in the sequence, are happening later in time. And again, this is all happening during odor B presentation. Looking at odor C and then D as well, I'm going to show only C for brevity's sake. We see a similar kind of dynamic playing out. Odor B is represented before the start of the trial. C is peaking at the beginning portion of the trial, corresponding to when we're training the model, so we know it's doing what it's supposed to. But then representations of upcoming items, in this case D, are present later on during the trial period. These findings are compelling in that they're demonstrating evidence that ensemble dynamics are capturing the sequential relationship among non-spatial events. Importantly, I have to stress this, is that these events are separated by multiple seconds in time. Whenever you see it saying that they're discontiguous, it means that there's a gap in between them. They're not happening one right after the other. There's multiple seconds in between. So now that we've looked at the task, looked at how there's temporal coding in the ensemble, trial-specific information, and this representational cycling among sequential events, we wanted to kind of test to see how far we could take this and see whether we could identify this kind of representational relationships within a single theta cycle. And so for this, I jumped over it. For this, we're using a somewhat simpler model, the multinomial logistic regression. And we're testing to see if there are non-spatial theta sequences similar to how there are spatial theta sequences, whereby activity along a segment of a trajectory in space, past locations are represented more on the descending phase. The present is decoded most in the trough of the theta cycle, and the ascending portion decodes mainly upcoming positions in space. So for this, we're looking at the first cycle within the trial period that starts 100 milliseconds after, that's starting 100 milliseconds after the start of the trial. We're thinking that this corresponds mainly based on the decoding and the behavioral data. This reflects a period when the animal is very attentive to the odor being present. And very briefly, what we find training on the trough of this cycle, we were expecting to find presentations of past odors in the descending phase, the present stimulus in the trough, and future odors in the ascending phase. And we see something very similar to that. Decoding of the present stimulus peaks in the trough. Past information is decoded at a greater rate than future information in the descending phase. And likewise, in the ascending phase, we see higher decoding of upcoming information relative to past. We see this also represented within the when we apply this model to individual tetraodes or individual cells. And we see something similar when we look across cycles to what we saw previously, whereby cycles before the start of the trial tend to represent the previous odors, and cycles that happen later in the trial represent future odors that are upcoming. So brief replay of events. Using our complex behavior and cutting-edge statistical approaches, we were able to identify novel forms of sequential organization within the hippocampal ensemble activity. We're replicating previous findings that temporal information bridges within and across events, that distinct trial relevant information is sequentially differentiated within these events as well. Most interestingly, the representations of items separated by multiple seconds in time seem to be cycling within within odors and events themselves, and that this representation seems to be able to be compressed within a data cycle. This seems to suggest a fundamental function of a network is to encode and preserve sequential order of experiences. And I figured the chicken would be getting the better of me, so just throw this up. Thanks to our collaborators and funding sources, and thanks for listening.